today's talk is by Dr. Kelly Ruggles. And she's a PhD from Columbia University in metabolic biology. She studied the genetics of type 2 diabetes and other lipid-related disorders there. And she has an undergraduate degree from Cornell in biological engineering. She's a postdoc with our faculty, Dr. David Fenyo, and she focuses on the development of informatics tools for proteomic analysis and for proteogenomic integration for the study of breast cancer biomarker discovery. And that's what we're talking about today. Thanks.
Thank you. Okay, great. So I'm just going to start with a little bit of background. So it's well known that cancer is a disease of the genome. And cancer patients acquire tumor-specific somatic variants, which can act as drivers of tumor progression and can also influence diagnosis, prognosis, and also treatment strategies. And for this reason, a lot of groups have focused on identifying and validating these somatic variants in different kinds of cancers. And when you do cancer genome sequencing, you have a lot of data. And there's been a lot of discussion in finding the meaning within this data. A lot of people focus on the somatic variants that occur most frequently. Others look at the key pathways that are affected by these mutations. But the data interpretation is still a major challenge. Also, there is a large amount of genomic heterogeneity. Whole genome sequencing has uncovered millions of germline variants between individuals. And many of these genomic studies typically use a reference genome in order to model the general population, which masks this patient-specific variation. So in order to address these challenges, we and other groups are using proteomics. And we're using proteomics to answer several questions. The first is that whether or not these genomic aberrations are detectable at a protein level. If they are detectable, what is their effect on the actual function of the protein? Also, can we use the proteomic information to classify cancer in perhaps a better way? Right now, we classify cancer using gene markers, which has actually been a very useful tool for classifying cancer. But perhaps proteomics can give even more information in this classification. Also, can we use proteomic information to guide biomarker development? And that's one of the main goals that I'll be discussing. And proteomics also provides us additional information that genomics cannot, including post-translational modifications such as phosphorylation status. And we can also use the proteome to further annotate the genome. Oops, sorry. So cancer proteogenomics. So proteogenomics means the intersection of genomics and proteomics. And we're using proteomic, genomic, and transcriptomic data from large-scale cancer studies to analyze in a patient-centric manner. So the patient-centric manner is in regards to this genetic heterogeneity that I discussed earlier. So we're taking into part all of the germline variants as well. And the goal of this study is to identify sensitive and specific biomarkers and to develop innovative methods for disease prevention and prognosis. So for these studies, we're working with two different consortiums. The first is the Clinical Proteomic Tumor Analysis Consortium, or CPTAC. And CPTAC is made up of many different institutions. We are working primarily with WashU and the Broad and UNC Chapel Hill, all focusing on the breast cancer, proteomics, and genomics. These other institutions focus, we work with them as well, but they focus on ovarian and colon cancer primarily. And the goal of CPTAC is to accelerate the understanding of cancer through the application of robust quantitative proteomic technology and workflows. So we also work with the TCGA, or the Cancer Genome Atlas, who's been working for many years doing the sequencing of cancer genomes. At this point, they've done thousands of cancer genome sequences. And their goal is to understand cancer through the genomic analysis. And so we're working with them to sort of combine the proteomic and genomics to get a better viewpoint of cancer biology. And we're working primarily with two different tumor types. Patient-derived xenografts, or PDX tumors. And they make patient-derived xenografts by taking primary breast tumors and injecting them into an immunodeficient mouse where they're able to grow. This provides us with hypothetically unlimited quantity of tumor, as long as you keep them growing on the mouse. And this is really important because then we can use this to assess drug resistance based on the genomic and proteomic makeup of the tumor. So you can have several mice with the same tumor, treat them, see how their survival changes and also how their proteome changes. So this is a really important tool for cancer biology. So these are being maintained at WashU. There are 45 PDX tumors total, 16 of which are undergoing proteome analysis that we're dealing with. In addition to this, there's the TCGA retrospective tumors. So as I mentioned, TCGA has spent many years doing genomic sequencing, and they have sequenced over 700 breast cancer tumors. And we are working with 95 of these tumors, which have passed quality control for proteome analysis, which is being completed at the Broad and WashU. So this is the overview of our study. We have, as I mentioned, the 16 PDX and 95 breast TCGA tumors. These have already undergone genomic sequencing and RNA sequencing. And we're currently doing proteomics at the Broad and WashU, both global and phosphoproteomics. So once we have the genomic and transcriptomic information as well as the proteome, we input all of this into a new pipeline that we've developed here called Quilts. And this takes in the genome and transcriptome. It identifies variants, novel expression, fusion genes, indels, copy number, and is able to create tumor-specific databases. And we then use these databases to identify tumor peptides within this proteomic analysis. And following this, we then do proteogenomic mapping and integration to identify biomarker candidates, to classify tumors, and to do downstream network analysis. So following this, we then come back with our biomarker candidates, and we come back to our original tumors, and then we do targeted proteomic studies on these candidates in order to verify the biomarkers. And I'll talk more about this step in the second half of the talk, so I'm just going to focus here on this new pipeline called Quilts. So the first thing that Quilts does is creates this tumor-specific database. So we start with a reference human database. In this case, we use Ensemble. And then we input the genomic sequencing germline variants from the non-tumor sample. We also identify alternative splicing, somatic variants, and novel expression using the genome sequencing and RNA sequencing of the tumor sample, and also put this into our tumor-specific database. And this is just an example of the specific novel peptides we're looking for. In terms of alternative splicing, we're looking for novel alternative splicing. So when you do RNA sequencing, you get this junction file where you can look for intron-exon boundaries. And we're identifying any spliced genes that are not within the reference genome. So any kind of novel alternative splicing. Also novel expression, where there's this new expression within a non-coding region, what was thought to be a non-coding region. We have fusion genes in which two genes from either different chromosomes or far apart within the same chromosome are fused together, and variants where there's a single nucleotide change. So we input all of this into our tumor-specific database. And now, before I go into how we identify the proteome using this database, I'm just going to go into some basics about protein identification and quantitation using mass spectrometry. So we have our tumor sample, which we then lyse and fractionate and digest, typically using trypsin, which gives us peptides. And then we inject these peptides into the LC mass spec, and we get a spectra. And we use the mass to charge, this is the x-axis, to do identification. And the way we do identification is by taking the spectra and comparing it to a spectral library, which can identify a similar pattern and give you the most probable sequence of this peptide. We can also use the spectra to get intensity. But the reason I'm mentioning this is because if you don't have your peptide within your spectral database, you're not going to identify it. So that's why we make these patient-specific databases, because in tumors, you're going to have peptides that don't occur in normal cells. And so if we just compare them to the normal spectra, we're not going to find them. So that's really the reason we make these tumor-specific databases. Okay, so now here we have our spectra from the proteomic analysis. And again, we do the peptide identification by matching the spectra with the peptide sequences in the sample-specific database. We then filter out anything that's normal, so anything that matches to the human reference genome. And we keep anything that's novel, so variants, alternative spliced peptides, novel expression and fusion genes. And we then use a tool which was developed by David and Manor Ashkenazi, which I'll discuss in detail in a little while, called PGX, which maps these peptides back onto the genome. And this gives us our tumor-specific peptide list. And this process of identification increases the sensitivity of identification compared to normal search engines. And it allows us to identify these novel tumor-specific peptides. So in order to validate our pipeline, we use 2 patient-derived xenograft breast tumors. They are from 2 different subtypes. They're basal and luminal. These they've already done the whole genome sequencing, RNA sequencing, and the global and phosphoproteomics on these 2 tumors. So we have all this information. So of course, we first created the tumor-specific library, and then we used 3 data sets of the proteome data sets from WashU, global proteome, to identify the variant peptides, novel alternative splicing, the peptides coded by unannotated genes, so this novel expression, and as well as fusion peptides. And then we tested visualization methods for the proteogenomic integration. So this is just looking at some of the specifics of these 2 tumor types. When we just look at the whole genome sequencing, we can see if we look at how many amino acids change because of germline and somatic mutations. You can see here that, so this is the number of amino acid changes within the protein, and this is the frequency of that occurring. And then the blue and the green are the germline for basal and luminal. So there are more germline mutations. That's consistent with what we know. Causing changes in amino acids, and then these are the somatic for the 2 tumors. So we found that 25% of proteins were affected by non-synonymous germline variants, and 2 to 3 were affected by somatic variants. When we compare this to our proteomic data, we do identify a small subset of these in our proteomics. So that was exciting. Additionally, when you look at alternative splicing, we can see, so here is the number of junctions and then the number of reads, and then we're matching them to either the reference genome or anything that doesn't match the reference genome. We classify either as being alternatively spliced, meaning that it is at an intron-exon boundary, but it's in a new way, or it's novel expression, which means it's within a region that was previously thought to be a non-coding region. So the blue is anything that matches the reference genome. The green is novel expression, and the red is alternative splicing, and this is reflected here by percentages. So as you can see, most of it matches known exon splicing. That makes sense. We have about 4% that are of novel alternative splicing and about 15% with novel expression. And we don't expect to see all of these reflected in our peptides. We expect to see a small percentage of them. And when we look at our proteomics, we do find that we do see some peptide evidence for these novel proteins. So at this point, we've made our tumor-specific databases. We've used the proteomics to identify these tumor peptides. We now move into the proteogenomic mapping and integration. So proteomic mapping, we map all of the different types of data, so the proteomic, genomic, and transcriptomic, to the same coordinate system. And then we integrate the proteogenomics by looking at not only at the changes in the protein sequence, but also the expression changes and also post-translational modifications within our data. And then we also want to observe this data from many samples at once and analyze them as a whole. So this is the mapping tool that I discussed previously, which was developed by David Fenyo and Manor Ashkenazi. It's web-based, so you can actually access it, and it's Python. It was written in Python, so it takes in a list of peptides, And you can either use the reference database or a sample-specific protein database. And it integrates the proteomic data onto the genomic coordinates and spits out a bed file and a bed graph file, which you can use in a visualization such as this, where like UCSC genome browser, this is IGV. You can just input this file in and you can actually see, so here is, I think this is chromosome 14, we're looking at AKT1 as an example. We have our somatic variants, our germline variants. This is the reference gene. This is the predicted alternative splicing based on our junction data, and then novel expression based on our junction data from our RNA sequencing. And then we can also map the peptides onto the same coordinates. So you can see what peptides are being expressed from what exons. And so it's a really useful tool. So we can also visualize some of our variant peptides. So here you can see this is a variant, a germline variant. And then the blue is a reference sequence, and the red is our tumor sequence. You can't see it here, but I've blown it up here. So you can see that there is an amino acid change that is consistent with this mutation. We can also look at novel alternative splicing. Here we have our reference gene in blue, our RNA sequencing in black. And as you can see, the RNA sequencing shows that it skips this exon here. And we identified a peptide that bridges these two exons, which is evidence of this skippage in the peptides as well. And then if we look, we can also see novel expression. So again, this is our reference. This is our RNA-seq data. And then here, the RNA-seq shows that there is expression in this intronic region, which we did find in a peptide form. So in addition to just looking at where the peptides fall within the genomic coordinates, we can also add in quantitative information. So here we've added protein quantitation levels, including phosphopeptide and just global peptide levels, and also the RNA-seq expression data. And something that we're working on incorporating into this pipeline is the cancer genome browser. And the cancer genome browser, this is what some of the output looks like. And actually, the cancer genome browser already has all of the TCGA breast cancer data within it. So this is the gene expression data from the breast TCGA. I just looked at 21 genes, but you can look at any gene you want. And it gives you this heat map that you can sort by different clinical features. They also have the RPPA, which is an antibody-based proteomic expression assay. There are, I think, probably a few hundred peptide proteins that they can measure in this way, but we can measure up to about 12,000 proteins. So we're hoping to make a map that looks something like this, except with way more information after we get all of the analysis completed. So just to summarize the Quilts pipeline, we've established this pipeline for the analysis, integration of the genomic, transcriptomic, and proteomic information. And we've tested the pipeline using 2 PDX breast tumor samples and identified the peptide expression for the somatic variants, alternative spliced forms, and novel expression. And we're able to map all of the omics information onto one coordinate system and include our quantitative information. So we're currently analyzing 95 TCGA breast tumors and 16 of the patient-derived xenografts using this tool. And we're going to use tools such as the cancer genome browser to add to this pipeline for this additional data integration once we have all of the proteomics integrated with the genomics. And then following the breast cancer analysis, we're going to move into both colon and ovarian cancer types. Okay, so I've told you all about Quilts. So I'm going to move in after. So after this is finished, we'll have hopefully a nice biomarker candidate list. And then we'll take this and we'll look at these proteins in a targeted approach. So we'll come back to the tumors and we'll do targeted proteomics. And so we're developing tools right now and assays right now to do this even before this part is finished. And this is being completed primarily at Wash U. And I'm going to focus in on all of the different steps. We have developed informatics tools at both the peptide selection step as well as the quantitation step. So far I've told you about global and phosphoproteomics, and most of the proteomics that I've discussed is considered shotgun proteomics. So after we lyse the sample and fractionate and do trypsin digestion, we inject into the LC-MS, and the MS1 records that mass to charge ratio and selects the peptides that are the most abundant. And it fragments those peptides, and then it measures those in MS2, and then uses this protein database search to identify peptides. In targeted mass spectrometry, you select your peptide before your experiment, and you measure that peptide, your precursor ion, then you fragment that peptide and you identify the peptide using this precursor fragment pair. And most of the targeted proteomics is, for biomarker discovery uses selected reaction monitoring, or SRM. So in SRM, you have your peptides, and you select you select your peptide in the first cell of your mass spec, and then it fragments that in a collision cell, and then it selects again for your fragment ion. And you use the precursor peptide and the fragment ion, which is called a transition, for identification. So this fragmentation, it actually breaks along the peptide backbone. In this case, the collision type we're using breaks at the peptide bond, and it's the Y and the B. So I'm going to be talking about Y and B ions, but and the number of the Y and B ion depend on where it breaks within the amino acid chain. And the way we quantify in SRM is we typically, and this is how we're doing it, most people do it this way, we use synthetic peptides, these heavy labeled peptides. So it's the same, chemically the same as the peptide you're interested in, but you've added in a C13 and N15 label, so you have a mass shift. And you add in a known quantity of your heavy, and then you compare your light to your heavy, and you can get your quantitation of your peptide of interest based on how much of your heavy you're seeing. So we use this peak area ratio, which is the light over heavy, and I'm going to come back to this at the end of the talk. So SRM has been used for biomarker discovery, where you have your disease and your controls, and you are interested in a certain protein or peptide, and you want to see if it's, let's say, higher in your disease population. You do the typical preparation for mass spec, and then you look and you compare how much of that peptide you see in your controls versus your disease population. And just as a motivating example, we're focusing on AKT1 and breast cancer, because our WashU group is really interested in the role of AKT1 in breast cancer and certain subtypes of breast cancer. So I'll refer to AKT1 a few times during the last part of this talk. So though selected reaction monitoring is a really wonderful targeted tool, there are some weaknesses. So here's just the method workflow of SRM. We select our protein, and then we select our peptide. And we have to select this peptide. So that is one sort of issue. You have to be careful about what peptide you select. We'll talk a bit about that. And then also you have to select your transitions, and you're limited in the number of transitions that you can measure. So you have to also be careful what transitions you select. And then you validate these transitions, and you sort of cycle through this, which can be really laborious and time-consuming and can be a bottleneck within the method. Following this, when you find good transitions that you can use to quantify, then you do your calibration curves, and you can actually find the level of peptide within your sample. So the WashU group that we're working with, as well as others, are using this new method called parallel reaction monitoring, or PRM. It's really similar to SRM, except so here in SRM you select your peptide, you fragment it, you select your fragment ion. In PRM, you still select your peptide and you fragment it, but you're able to measure all of the fragment ions. So you don't have to choose your transitions in this case. And you can use all of these transitions to confirm your peptide ID and also to measure your peptide quantity. So we've been working very closely with the group at WashU, and we've come up with we've been able to help them at 2 different steps of this process. The first is in the selection of peptides, and the second is in the selection and validation of transitions post data acquisition. So here in PRM, you can see we're able to skip this selection of transition step and just measure everything and then select and validate after we measure all of the transitions. So I'll start by going through the tool we developed for selecting peptides. So the peptides that we select have to be proteotypic, meaning that they only map to one gene, and also consistently observed by other mass spec methods. And this is the workflow. I'm going to go step by step through it. But this whole process is fully automated and can take either a list of proteins or peptides as input. So we first start here, if we start with a protein sequence in FASTA format, we do an in silico trypsin digestion and gives us peptides. Oops. We then compare these peptides to the human reference database to find which peptides and genes correspond to each peptide. And we really are only interested in the peptides that correspond to only one gene. We then compare our peptides to the global proteome machine database, which is a really useful tool. It was constructed to aid in the validation of tandem mass spec and protein coverage patterns. So it allows users to compare their results with other previously observed results, and it contains many, many peptide records. So what happens is when somebody publishes a paper with mass spec data, GPMDB goes in and does a quality control step. If it passes that, it takes it in and then it stores it. So you're able to input your peptide and see how many times other people have found that spectra or peptide. So if you input your peptide and you don't see that anyone's seen it, you should not use it because it's probably not a good peptide. It's either too hydrophobic or has some sort of other issue. So we then find out how many times the peptide's been observed. And we also compare it to the CPTAC mass spec data. So this is the consortium we're working with, just because they're working with other tumor tissues, and we want to see if they've seen a similar thing. And this gives us our final list, which is genes, sequence, whether or not it's proteotypic in different backgrounds, and how many times it's been found in GPMDB. So after this step, they're able to run the mass spec and get the transitions, at which point we can select and validate these transitions for quantitation. So the main issue with doing this, with looking at transitions for quantitation, is that you have to take into account interference. So interference occurs when a peptide has a similar mass to charge and chemical characteristics as the peptide of interest. So if your blue is your peptide of interest and your orange is this interfering peptide, let's say it gets into the first cell and then it is fragmented in your collision cell. And then it may have also a fragment ion that is similar to the fragment ion that you want to study. So if you look at the spectra, you're going to have some sort of interference in some of your transitions, and you have to really be able to pick out which transitions are good to use for quantitation. So the way that the lab at WashU has been doing their PRM is they do all of the light and the heavy peptide in both simple and complex matrix. The simple matrix is a peptide carrier solution, and the complex matrix is an unfractionated tumor digest. So it's a really, really complex solution. And the idea is that the simple matrix should have a minimal interference, and we use this as a reference. And the relative intensities of these transitions should remain constant within all of the spectra. So in the simple, let's say this is Y2, this is Y3. Y2 should always be larger than Y3, and then this is Y4. It should all be relative. And the simple is our reference. Then we can look at our complex and we can see, are those relative intensities the same? And if they're not the same, there's likely some sort of interference. So here's a really simple example of how our algorithm works. We do this for both light and heavy separately, and then we bring them together at the end. So if we're just looking at light, we have our simple and complex matrix. I just picked out 3 transitions and the intensities of each of these transitions, which are shown here. So we then make a ratio of transitions for each spectra. So this is the relative intensities. So for each transition, we make a ratio to each other. And we do this again for simple and complex. And our simple is our reference. So anything that's really different in the complex matrix indicates that there is some sort of interference. So if you just look at this, so here, this 0.6 is fine, 1.67 is fine, but all of the the ratios that have Y5 are off. So this indicates that Y5 has some sort of interference. So you can do this easily by eye when you have very few transitions that you're looking at, but when we automate this, we just simply took the complex over the simple matrix, and anything that's close to 1 indicates that there is very little interference, and you can set a threshold. So we set a threshold, and then anything that's outside of that threshold indicates that there's some sort of interference occurring. And so this is just how the automation works. So again, we have light and heavy, simple and complex for both. We make branching ratios for each of these spectra, and then do complex over simple. And then this is just a way of visualizing the data where anything that meets our threshold is in red, and anything that does not meet it is in white. So like for instance here, Y9 is all white. It's clearly has some sort of interference. And we have an algorithm that then goes through and picks out the good transitions. And so this is the workflow. I've gotten you to this point. We then have a good and bad transition list, and we can rank these based on their mean ratios, and then use this information to quantify. And I'm going to go into a little bit of detail about that because it gets very confusing. So let me remind you that we use these synthetic peptides, the heavy peptides, to measure our peptide of interest. So we spike these in, and we know the concentration we're spiking in, right? And then we make a light over heavy ratio to get our peak area ratio, and we can use this to determine how much of our peptide of interest is within our sample. So the way we do this when we sort of combine all of our transition information is we take the heavy and light transitions and we find a ranking for them based on both of their rankings. So we average their rankings and we get like an end rank. So this is the transition ranking in terms of good transition to bad transition. So if there's interference in both the heavy and the light for one, it's going to be really bad. And if there's no interference in the heavy and light for one, it's going to be really good. And then there's sort of an intermediate within that range. So we then, as I mentioned, we want to get light over heavy. So we take our best transition and we calculate light over heavy. So in this case, we're only looking at the complex. We don't care about the simple. We're just looking at our tumor matrix. So this is the first point, light over heavy. Then we add in our next best, and we again calculate light over heavy. And we continue doing this successive summation, and we can set a second threshold where we can say, okay, we don't want our quantitation error to be more than 20%, which is what this threshold is. So here, this is the last transition that is within that threshold. So we can say, in this case, use these 3 transitions to quantify your data, and you won't have an error outside of this certain percentage. In some cases, all of them are fine. I've seen that. So it just depends on the data that you're looking at. So the last method I'll discuss is called swath mass spec, and it's really a new method for targeted peptide quantitation. So it uses a high-resolution instrument, and instead of choosing that peptide in that first step, it just lets in 25 Dalton isolation windows. So it lets in a lot more potential interferences, but you can measure a lot more data at once. And it generates high fragment ion spectra for all precursors within that window. So it's the same workflow as the parallel reaction monitoring. But this step becomes even more important because you have a lot more potential interferences. So this is what the data looks like. We're still working on this. They're still figuring out the best way to do this. But as you can see, if we set our threshold here, we have, in this case, we're looking at way more transitions because we are added neutral losses. But we only have 2 transitions that are considered to be good in this case because this is such a complicated spectra. But if we can get this to work, I think it'll be really great because we can do so many more peptides at once. So just to summarize the targeted proteomics section of the talk, we've developed 2 informatics tools to aid in the proteomic analysis for accurate peptide quantitation. The first is in peptide selection, where we select proteotypic peptides that have also been found in other LC-MS methods. And we also have developed this algorithm to identify transitions with low interference. And we're currently developing assays against 500 potential biomarker peptides, which we'll use in the TCGA and PDX samples once we've gotten this working, which it's close. And we're also using, as I mentioned, we're going to use the target assays to validate these in the TCGA tumors. So this is, again, the flow. And we are we're done with this, and this is still going. And we've finished most of the actual designing of this pipeline. And we're still working on this. So we're just waiting for this information to sort of flow in. And I'm sure we'll run into things we'll have to fix along the way. But I think we're pretty close. So with that, I'd like to thank everyone in the Fenyo lab, especially, of course, David, and all of the other people at NYU who've really helped with this project. Both Stratos and Eric at the High Performance Computing Facility, who I'm sure are going to help us a whole lot more in the near future with the massive influx of data. And then Manor, who really made this PGX, which is a wonderful tool that we've been using a lot. And then the groups at WashU, Reed Towson and his lab, are the people who do all of the targeted analysis. So we have at least 2 meetings with them a week to discuss all of this informatics work. Matthew Ellis, who is the primary MD on the project, the breast cancer project, and of course the Genome Institute, who were really involved in the TCGA genomic sequencing and who are now helping us develop our pipeline because they developed their own pipeline for the genomic analysis. And then the Broad and Steve Carr's group, who are doing most of the global and phosphoproteomics of the TCGA samples. UNC, who are also doing phosphoproteomics for some of the PDX samples. Sam Payne at PNNL, who helps us with the proteogenomics. And of course, Karen, who also helps us with the computing issues. So thank you. Anybody have any questions?
So I know you just started looking at the SWOT data, but how is that promising, or does it look like, is there a lot more interference?
Right now, I think it's not great. I'm waiting for them to send the next. This is not high resolution, so so when they send the high resolution, if it looks like this, I don't think it's promising. If it
still up in there, it might be, these samples might be too complex for that.
Yeah. So we'll see. It would be great if we could figure it out, but yeah.
I have a question not related to the talk per se, but just more from the scholarly student perspective. How did you get interested in proteomics and genomics and breast cancer?
So I got interested in proteomics when I graduated from college. I actually worked in a mass spec lab. And then when I was at Columbia, I did almost all genomics work. So I think this is a really nice sort of combination of my backgrounds. In terms of breast cancer, I think I'm really interested in it, but that was not the primary I think that was just now I really like what I'm doing, but it wasn't. I'm more interested, I think, in the proteomic, genomic integration. Are we good otherwise? Okay, thank you.